Welcome to our channel Easybiotech. In this channel, we try to simplify the biotechnology topics for easy understanding. The students who are studying biotechnology or any person who is interested in biotechnological topics, they can check our videos and understand easily each and every topic. So let's begin. Today the topic on which we are going to present is replication. First of all, we start with the definition of replication. What is replication? Replication is a process through which DNA can make exact copies of itself. It can also be said as it is a biological process by which a double-stranded DNA molecule is copied to produce two identical DNA molecules. It is a semi-conservative process in which each of the two double helix form the parent double strand have one old and one new strand. Below is the figure which simplifies this definition. Here is the original DNA molecule which has been replicated or copied to form two different DNA molecules same like itself. What is a DNA? DNA is a nucleic acid which is present in all living organisms, whether plants, animals or viruses. Here in this figure you can see this is the cell in which there is a nucleus, and in nucleus contains chromosomes, and in the chromosomes is present the DNA. DNA is found in the chromosome. DNA molecule is a polymer consisting of several thousand pairs of nucleotide monomers. This figure shows below it clearly. Each DNA Where does the replication takes place? Replication takes place during the interface between two mitotic cycles. What are the things which are needed for replication? Replication needs a DNA template, see here, which is nothing but a single-stranded DNA, a primer here, a deoxyribonucleoside triphosphates, magnesium, DNA, unwinding protein, Superhelix relaxing protein, a modified DNA polymerase to synthesize the RNA primer, the product of some genes, and polynucleotide ligase, which is a joining enzyme. Replication begins at a specific point called the origin. Here you can see in this figure. One model suggests that replication starts with a nick or incision made by an incision enzyme endonuclease. Here in this figure you can see the nick. Here one nick is there. It is here the nick is there, and here this nick is there. The two strands of DNA double helix unwind with the aid of a DNA unwinding protein, also called the DNA binding protein, which binds to a single DNA strand. Here you can see the double DNA strand has been unwinded to single strands. The strain imposed during the unwinding of the strand is revealed or relieved by the action of superhelix relaxing protein. Initiation of DNA synthesis needs an RNA primer which is synthesized by DNA template close to the origin of replication. This synthesis is catalyzed by a special form of RNA polymerase. Deoxyribose nucleotides are now added to the end of RNA primer, and the main DNA strand is synthesized on the DNA template, which is complementary to the DNA strand and is synthesized by DNA polymerase. Here you can see this is the DNA template to which the RNA primer has added. Now the new nucleotides will go on adding and double strand would be forming. Polymerase 1 now degrades the RNA primer and simultaneously catalyzes the synthesis of a short DNA segment to replace the primer. This segment is then joined to the main DNA strand by a DNA ligase. Here in this figure, you can see how the DNA polymerase is manufacturing the new strands and the double strands are being formed. Replication takes place discontinuously and short pieces called Okazaki fragments are synthesized. One strand may synthesize a continuous strand and the other Okazaki fragments, or both strands may synthesize Okazaki fragments. Both new strands are synthesized in 5' to 3' direction. Thus, one strand is synthesized forward and the other backward. The Okazaki pieces are joined by polynucleotide ligase, a joining enzyme, to form continuous strands. Replication may be in one direction, that is unidirectional, from one point of origin, or in both directions, that is bidirectional. In this figure, you can see here a continuous strand has been formed, whereas here Okazaki fragments are formed. Now these Okazaki fragments, which are discontinuous, will be joined by the enzymes. Now we will move towards the mechanism of replication. The replication mechanism involves the following points: DNA synthesis with genes, initiation starts replication, origin point, strand unwinding, superhelix relaxing protein, DNA template, RNA primer, chain elongation, replication has direction, Okazaki fragments, and polynucleotide ligase. Process of replication requires the products of genes. Here we have took the example of E. coli. Below are the genes which have different functions in E. coli, which helps in replication. For example, gene DNA A and CD helps in initiating new rounds of replication, whereas gene B and D helps in the propagation of replication. Pol A and Pol B are responsible for polymerase 1 and 2. Pol E helps for the production of polymerase 3 and 3'. Origin gene helps in origin of replication, and terminase helps in terminase of replication. Now, initiation starts with replication. Initiation enzyme endonuclease makes a break or a nick in one of the two parent strands, which starts replicating. Here you can see this enzyme is making a nick. Origin, a new site on the chromosome for starting of replication. Here we have took the example of E. coli. E. coli has its origin at 74 minutes on the chromosome. Here you can see in the image. DNA unwinding protein binds preferably to single strand of DNA and promotes unwinding of double helix DNA. The region of double helix where the protein binds a Y-shaped replication fork is formed. Here you can see very clearly in the figure below. Superhelix relaxing proteins. The strain imposed by unwinding is relieved by the action of a superhelix relaxing protein. The protein has a nick closing enzyme activity. By introducing nick in the non-replicating region of DNA, one strand is made to rotate upon the other, thus relieving the strain. The break is then closed by ligase and the replication fork moves on. You can understand this with the below figure. DNA synthesis requires 4 deoxyribonucleoside triphosphates and a DNA template. The previously existing DNA on which a new DNA is synthesized is called DNA template. Here you can see, as the DNA is unwinding, this single strand here in blue color and here this is in green color acts as the template, and the nucleotides get added to these and form the double-stranded. Initiation of DNA synthesis requires an RNA primer. A pre-existing polynucleotide chain is required, to which polynucleotides are added. The primer RNA is a short polynucleotide chain with a 5' triphosphate end and a 3' OH end. Here you can see this is the DNA template to which an RNA polymerase with 5' has been attached. This is required. RNA primer is required because to this RNA primer now the new nucleotides will get added and the chain will start. Chain elongation. Synthesis of new DNA strand takes place by addition of DNA nucleotides to 3' OH group of the last ribonucleotide of the DNA primer. This synthesis takes place in 5 to 3 direction and is probably catalyzed by DNA polymerase 3. The newly synthesized DNA chain has a primer RNA attached to their 5' end. The RNA primer is hydrolyzed by the 5' to 3' exonuclease activity of DNA polymerase 1. Here you can see below in this figure clearly how the primer are there and how the DNA polymerase just adds the nucleotides and the new strands are formed. The resulting gap is filled by the DNA nucleotides, again by the catalytic activity of DNA polymerase 1. The newly made DNA ID joined by DNA ligase to form higher molecular weight DNA RNA primer, which are joined on both sides of DNA chain, are digested by specific endonucleases. Here the below figure shows this very clearly. Now replication has direction. Replication may be in one or both direction from the point of origin. Replication in one direction is called unidirectional replication, while that in both direction is called bidirectional replication. Here you can see one strand is formed continuously but in one direction and the second strand is also formed. So here both the strands are formed simultaneously. Replicating forks. At a point where the two strands are separated by replicating fork is formed. The fork appears in the form of a Y and advances during replication. In bidirectional replication, the separated strands between the two forks appear as a bulb or an eye. The speed or extent of movement of the two forks in opposite direction need not be same. Here you can see how this Y or replicating fork is formed. Okazaki fragments. During replication, only one strand, the 3' to 5' or continuous strand, is continuously replicated. This other strand, that is 5' to 3', replicates in a discontinuous manner, synthesizing short fragments which have been called Okazaki fragment. In the below figure you can see one strand is continuously formed, whereas this other strand, it is formed in discontinuous way and it is called Okazaki. Polynucleotide ligase. Replication is a discontinuous process and takes place in short segments called Okazaki fragments. Now, polynucleotide ligase joins these fragments into higher molecular weight DNA. Here you can see the Okazaki fragments are joined by the specific enzyme to make a continuous fragment. So viewers, we hope that these slides have helped you in understanding the process of replication in an easy manner. We would be waiting for your comments and suggestions. We would be very happy to get your mail at easygobiotech@gmail.com. If you like our presentation, then please do like, subscribe and share our channel. Thanks for watching.